All right, welcome back. In this video lecture, I am covering 13.2, the structure of DNA. So I'm going to talk about the chemical components of DNA, describe clues that helped us determine the structure of DNA, and then explain what the double helix model shows about DNA. So DNA stands for deoxyribose nucleic acids. Okay, so deoxyribose nucleic acids. And way back in the beginning of the year, we talked about 4 macromolecules of life: proteins, lipids, carbs, and nucleic acids. And each of those macromolecules, macro means big, macromolecules of life are made up of smaller subunits called monomers. Like lipids are made up of fatty acids. Carbohydrates are made up of sugar molecules or saccharides, right? Monosaccharides. Proteins, what are proteins made up of? They're made up of amino acids. And nucleic acids are made up of nucleotides. So the monomer or smaller subunits of a nucleic acid is a nucleotide. And these nucleotides join together by covalent bonds where electrons are being shared. So nucleic acids are made up of building blocks called nucleotides. Now each nucleotide has 3 parts to it. Okay, a phosphate group, a sugar, in this case with DNA it's called deoxyribose, and then a nitrogen base. And there are 4 different nitrogen bases that we'll look at in DNA: adenine, thymine, cytosine, and guanine. But these are the 3 components of a nucleotide. Now, the nitrogen bases, there are 4 of them, and you may remember from Life Science 7, ATCG. Okay, so we got thymine, or abbreviated as T, cytosine as C, adenine A, and guanine G. And I just want to talk a little bit about the structures here. So here we have our our phosphates and our sugar group, and then these are the nitrogen bases. So the sugar and the phosphates are the same for all of the 4 nucleotides, but the nitrogen bases, that's what makes them different. And you'll notice that A and G, adenine and guanine, are double ringed. Okay, and so a nucleotide, okay, a nitrogen base that is double ringed, is considered something called a purine. Okay, and so a little mnemonic saying to help you remember that adenine and guanine are purines, as you could say all gods are pure, even though, you know, whatever you believe on that, but it's just a little saying there, all gods are pure. Cytosine and thymine, C and T, you'll notice that they are single ring, and we call them pyrimidines. Okay, now I will come back to pyrimidines and purines when I discuss a little bit about Chargaff's rules. Now the nucleic acid structure, as you can see here, we have covalent bonds between the sugar phosphates. Okay, so we have a kind of a single, like 1/2 of a ladder going on here. So here's a purine bonded to a pyrimidine. So what forms the backbone of a DNA chain? Well, this is what I mean by the backbone. It's the sugar phosphate. This would be the rung, because you're going to learn here that DNA is kind of like a spiral staircase or a twisted ladder. All right, so the hunt was on. Okay, after Avery Griffith's experiment and Hershey and Chase confirmed that DNA was the transforming principle, people wanted to know, what does DNA look like? Okay, because it's a really, really small molecule, but no one had any idea what it looked like. They could figure out chemical composition of it, but they couldn't figure out how those atoms were arranged. And so Chargaff, I think his name was Edwin, discovered that the same 4 bases found in DNA are found in all organisms. Okay, so in bacteria, plants, all animals, they have the same 4 nucleotides, A and T, C and G. And he also noticed that they were found in, that the proportion differed from organism to organism, but one commonality that he saw was that no matter how much adenine you had, you always had the same or roughly the same amount of thymine. And no matter how much cytosine or guanine you had, they equaled the other nucleotide. So here's the source of DNA from 4 different species: Streptococcus, which is a bacteria; yeast, a fish, heron, and a human. And here's my 4 nucleotides: adenine, thymine, guanine, and cytosine, and the percentages that it was made up of. So you notice that A and T, these values are pretty close to each other, okay, especially in the yeast here, and the heron, and the human. Notice that G and C are roughly very close to each other. Okay, so Chargaff's rule states that adenine equals thymine, cytosine equals guanine. Okay, so you started to see these patterns. Rosalind Franklin was studying DNA by trying to take a picture with X-ray crystallography. I think I said that word wrong. Sorry, it's really late at night right now. But she was taking pictures of DNA using X-ray diffraction, and she snapped this famous photo, depicted here as photo 51, and her results provided additional clues to what the DNA structure looked like. First off, you start to see this X-like pattern, and you have to envision looking down a DNA or a twisted staircase and seeing those nucleotide nitrogen bases inside that molecule. And so that this molecule right here was likely 2 strands. So you're kind of like looking down and it's bridged. I mean, it's just a really awesome picture. So DNA is likely 2 strands. The nitrogen bases were found inside the molecule. It's like a coil of springs, or, you know, a double helix is what we call it. Watson and Crick are credited for discovering the structure of DNA. Now, they didn't do any research on their own. They didn't do their own experiments. What they did is they collected evidence from different scientists and then compiled it and then kind of put it all together. So they were, you know, like each scientist, you know, like Rosalind Franklin and Chargaff and Linus Pauling and a few others, they were like pieces of a puzzle, and these guys, you know, just kind of put it together. So James Watson, depicted here, and Francis Crick were trying to figure out the structure of DNA by building models. And in 1953, Watson was shown a copy of Franklin's X-ray photograph, I believe, at a cocktail party. And he was like, Eureka! This picture right here, I think I know how it all works. And so they built this model out of metal and wood. You can see ring stands and clamps holding the sugar phosphates and the nitrogen bases and just showing how it works. And they were able to explain the specific patterns and properties of DNA. They could explain Chargaff's rules, and they could explain a few other things that they noticed from different experiments. So we call it the double helix model, or a twisted ladder. It explained Chargaff's rules because A, this nitrogen base pairs with T on the other side. So you can see DNA is made up of 2 strands, and they match complementary to each other. These nitrogen bases are held together by hydrogen bonds, and they're very, very specific. That is why C equals G, because C only pairs with G, and T equals A, because T only pairs with A. So we have covalent bonds holding the sugar phosphate backbone, but what's really holding the nitrogen bases together, kind of like one half of a zipper, are hydrogen bases. DNA does have direction, and it is anti-parallel. So if you imagine the 2 halves of DNA, they just run opposite to each other. That's all anti-parallel means. And so this allows the nitrogen bases to come in close contact with each other and to carry a sequence of nucleotides. Now I'm going to go back to this picture right here, and I'm going to draw and see if this works. Okay, so Chargaff's rule states that A equals T. So that means A pairs with T. So here I have A, and T, you know, would be here. A pairs with T. Okay. How many rings do you have coming across the rung of this ladder? So you say 1, 2, 3. 3 rungs. Okay, or sorry, 3 rings. All right, and then Chargaff's rule also states that C equals G, right? So here I have G, and all right, it's going to bond with C, a pyrimidine, single ring. How many rings do I have going across? 1, 2, 3. Now, if Chargaff's rule was not true, let's say C pairs with A. No, not A, sorry, T. So here's C paired with T. How many rings do I have going across now? 2. So you notice that I have this nice like ladder thing going on, and then all of a sudden it gets, well, that's not what I wanted. Erase that. This is what I wanted to do. It gets narrow, okay? And let's say I pair this with a C as well. Narrow. Right, it gets narrow, and then, you know, if I were to continue, maybe I have 3 rings going across. Well, then the DNA would come out again, right? So a pyrimidine has to bond with a pyrimine to maintain the same number of rings going across so that we have the same diameter for a ladder. Because if it didn't, I mean, DNA would be all wonky. You have some parts that would be super narrow, some parts that would be super wide. I mean, could you imagine two purines being bonded across? Like if I had another ring, then, you know, DNA would pop way out. Well, that's not the case. So that kind of explains why A pairs with T. A pairs with T, a purine with a pyrimidine. C pairs with G, a pyrimidine with a purine. Okay. So what holds the nitrogen bases together? Hydrogen bonds. Okay, these are very weak bonds, but because there are so many hydrogen bonds, the DNA molecule has a lot of nucleotides, it's pretty strong. Okay, so A pairs with T, C pairs with G. We get some base pairing or complementary pairing, and this base pairing explains Chargaff's rules of A equals T and C equals G. So you can see in here, now we got a nice uniform backbone, same length of rungs going across. All right, so that does it for 13.2, structure of DNA.